Good morning, everyone. Hope you can hear us. Welcome to our inaugural webinar from Collaborations Pharmaceuticals. I have with me Kim Zorn and myself. Thank you for joining us both today to learn about Assay Central and how we at Collaborations Pharmaceuticals can help you with your machine learning projects. Just a few logistical points today before we kick off. We would be very happy to take any questions you have, so please put them in the Q&A box at the top of the screen. You can see the icon here on the slide. You can also send us any messages privately as well using the messaging function that should be visible on the screen. And we will answer any questions that you may have at the end of the presentation. I'd like to begin today by sharing a little bit of brief information on Collaborations Pharmaceuticals and both Kim and myself, for those of you that don't know who we are. I'll also share a little bit about some of the types of projects that we do and also try to give you some outline of the types of projects and work that we have been working on in the last 5 or so years. Most of these have been enabled by our machine learning approaches that we're focusing on today. And Kim's going to give you a deeper overview of the technology and briefly demonstrate Assay Central. We will also try to keep this pretty brief, so if there are any other additional questions or things you'd like to see, we'd be happy to set up individual calls later. And finally, we're going to summarize some use cases that we've been doing in terms of some of the projects and examples we've worked on and how we may be able to help you and your companies and institutes in the types of projects and problems that you may be having. So a little bit about Collaborations Pharmaceuticals. I'm Sean Eakins. I'm the CEO of the company and founder. I have over 24 years of experience of working on machine learning projects. I've been working in this area at large pharma, small pharma, and I've been involved in starting a couple of companies, and this is the second company that I've started. We founded the company in 2015, and we're currently located on the NC State University campus here in Raleigh, North Carolina, and we have 10 agile employees and 3 labs here on the campus. We've been able to work on quite a wide variety of projects, mostly on infectious, neglected, and rare diseases. We've been able to file a number of patents on new molecule IP. In addition, we've also filed 8 orphan drug designations and been awarded them by the FDA, as well as a rare disease designation. So this slide gives a little bit more of an overview of the types of things that we do. We really deploy our machine learning algorithms alongside our deep drug discovery expertise to help in different projects. Ideally, we're working on small molecule projects for rare and neglected diseases, as I mentioned. And we also provide fee-for-service consulting services to other companies. Our sweet spot really is early hit-to-lead optimization. And that's where we've put a lot of effort. And we generally, with many of these collaborations and projects, we generally address tough challenges related to ADME tox, as well as involved in curation of data sets that our clients may need. And we'll give a little bit more information on that later on in the presentation. In the process of the company being formed, we've been able to build a vast array of thousands of models and data sets relating to diseases, relating to targets, and also physicochemical properties. And these can be obviously used to screen libraries of molecules and select compounds for testing in vitro or in vivo. We've also used our machine learning approaches to model in vivo data. Most recently, we've been developing software tools to assess synthetic feasibility with some of our collaborators, and I'll mention these later on as well in a couple of slides. The image on this slide gives a little bit of a high-level overview of our general philosophy. We focus primarily on curating the data, making sure that it's clean to begin with, and then moving on to building models, validating them, generating predictions with new molecules, and then obviously performing experiments and feeding the data back in. And this is something that we've done internally with projects as well as externally with clients. So I would say that none of this really would be possible without the funding we've been able to receive from the NIH as well as the DoD and North Carolina Department of Commerce. These have all been essential for us in building the software and also advancing many of the projects that we work on. In particular, the NIH, NIGMS, their funding has helped us build Assay Central software that I'm going to focus on today, and Kim's going to present. So this is a big thank you slide for all the funding to anyone from the NIH or the DoD that's watching us today. So by way of introduction, I'd like to introduce Kimberly Zorn. She's a senior scientist with us at the company, and she joined us just over 3 years ago. And she's been really instrumental in helping to shape Assay Central and work on many of the different types of projects with collaborators, both academic and also industrial clients. She manages currently a team of 2 other scientists that work with her on the different projects. And she's been able to present very widely, both at the American Chemical Society as well as other conferences in the last few years on the types of projects that she's worked on. She's been able to publish 12 papers using Assay Central in this short period of time. And we have several others that are currently in submission as well. Kim is quite energetic, so I'll just give you a heads up before she begins. And I'd like to hand it over to you, Kim.
All right, I'm going to do my best to talk as slowly as possible, but everyone feel free to jump in if I'm getting too fast and ahead of myself. So before getting started into more of the machine learning side, I did want to just do an overview of the available collections we have of machine learning models and data sets, including Assay Central, which is our core technology that we use to curate data as well as build and validate our machine learning models. But we also have Megatox, which is a subset of toxicology targets and molecular property models, as well as Megatrans, which is a subset of human drug transporter models. And we also have Megapredict, which is where we utilize thousands of chemical data sets to find new potential targets for new molecules. So first things first, what on earth do I mean by machine learning? Well, in the simplest way I can put it, it's what some experimentalists can do after decades of experience, and that is finding chemical patterns in biological data. But we utilize a computer to do it in minutes. First, we do this by assigning molecular descriptors to, as the term implies, describe the chemical features that are present in a data set. Then we identify which of these features are present in active and inactive compounds. And finally, we apply this knowledge to new molecules to predict target bioactivity. Now, one of the first things that I'm asked by new collaborators is, what kind of data do you need? And usually they are very pleasantly surprised that we only need 2 things, really. And that is standardized parent structures. And what I mean by that is desalted, neutralized, showing as much stereochemistry as possible, and a corresponding activity to go with it. Most often we see this in concentration units like micromolar or nanomolar, but we've also worked with other units as well. We've gathered large data sets from public sources like Chembl and PubChem, but we've also worked with small unpublished data sets from collaborators that are more focused on their needs. But I also always like to ask some bigger picture questions of any experimentalists that are involved in a project. Namely, is there a reasonable threshold for defining activity that we should be aware of? Because, for example, while 10 micromolar might sound like a great target activity to some, to others, they're really looking for more of a picomolar activity. Also, is the data that we're utilizing predicting purely binding to a receptor, or is it a downstream agonist effect, for example? And these all feed into how you, the user, are going to be utilizing these models and predictions. And while these questions are not essential to creating the models themselves, they help us determine how to best serve you with the tools at our disposal. So now that we've put together a training set, that's what we use to build or train our model, we have to, as I previously said, evaluate molecular descriptors. Now, there are all types and flavors of molecular descriptors. They can be things like rotatable bonds or hydrogen bond donors or acceptors. But Assay Central specifically uses extended connectivity fingerprints of maximum diameter 6, ECFP6 for short. And think of this as starting at one atom and iteratively working your way out to identify up to 6 atoms in radius. And we do this for all of the molecules present in a data set. Then the machine identifies the fingerprints that are comprising the active and inactive molecules, and the ratio of the fingerprint I in active compounds is termed its contribution. Once this information is collected, a Bayesian algorithm can be applied to the data. And without going into too much detail, the Bayes theorem is used to describe the probability of an event, in our case, bioactivity at a target of interest. Assay Central models also include a series of internal predictive performance metrics to give the user confidence in the dataset, and we have an example shown here at the left. Specifically, we use 5-fold cross-validation to generate these metrics. And what I mean by that is we take the total training set and randomly split it into 80% and 20%, being our temporary training and testing sets. Then that 80% predicts the 20% and checked for accuracy, and then it's repeated another 4 times so that we get a total of 5 rounds. So once we have a model generated, we are applying it to predict external molecules. We do this by first evaluating the fingerprints that are present in the molecule being proposed, and then the probability-like prediction score that is assigned to it is the result of summing the contributions of these fingerprints that are present in the training model. Now, usually these scores will fall between 0 and 1, so we always like to start that a score that's above 0.5 is what is predicted as active. Additionally, each molecule, when it's assigned a prediction score, also is assigned an applicability score, which provides a measure of how well represented the prospective molecule is in relation to the training data and can give you more confidence in that prediction. We regularly generate a variety of prediction types for large commercial screening libraries, but say you have a series of molecules you want to prioritize for synthesis, for example. Well, our visualization features within Assay Central can assist you in doing so. Here we have 2 molecules, and between them 2 central circles. Now, within these little circles are representations of 3 Bayesian prediction and applicability scores. The color of each pie slice is reflecting a prediction score from a target, off-target, or property category model, and the radius of the pie slice corresponds to the applicability score. Predictions of target bioactivity are presented on the left, and in this case, green is a desirable feature because it's an active feature. On the right is an off-target bioactivity scheme, where blue is a desirable feature because it is an inactive feature. You can use atom coloring to direct your syntheses by iteratively changing the molecules within an Assay Central bundle and observing the predictive effects quickly and seamlessly. In addition to the applicability scores, you can further investigate the similarity of your molecules in relation to the training data with these honeycomb plots. Herein, the proposed molecules from our previous slide are presented in the black background hexagons, while the training molecules are shown in the white background hexagons. And those that are highlighted green were considered active in the model. These plots are organized so that increasing distance from the central proposed molecule that this whole plot is built around corresponds to decreasing similarity to that molecule. So otherwise, you can use this to explore what has already been tested and how potential compounds can relate to the known chemistries within the training data. Another form of predictions and visualizations is in the form of prescriptions. And this is a method to score many types of models all at the same time, while defining what desirable and undesirable activity is. So for example, what I have shown here, we are only desiring activity against GSK3 beta in our target category. And we are not desiring activity against 2 cytochrome P450s and GSK3 alpha in our off-target category. Here, each box is corresponding to a model and is colored according to the prediction score. from the same category scheme that you saw previously. But here, the black bars beneath each square are reflecting the applicability score. Also, in this case, you'll notice that if a compound is present in the model, it's indicated with a black triangle in that box. And rather than having a prediction score, it's just a true-false statement of activity according to that model. In prescriptions, the higher consolidated the score, the more ideally the compound scored against the evaluated models. These prescriptions can be distributed within a set of model bundles to a collaborator, as I said, who can then run in vitro tests and enter edit mode within the prescription to actually input the results that they've generated. And this is really handy because then they can download this in a machine learning ready format, deliver it back to us so that we can complete a feedback loop. As Sean already stated, the Assay Central workflow is relatively simple. Curate data, build models, generate predictions, perform experiments, and then integrate the new data into a new model iteration and start all over again generating new predictions. We have completed this feedback loop with both external collaborators as well as several of our own internal projects so that we're constantly improving our predictions. With my last slide, I will direct you to assaycentral.org, which is a publicly accessible set of a variety of models. These include disease targets like Ebola, host targets like HERG, as well as molecular properties like cell permeability. And due to the ongoing pandemic, we also have COVID-19 related models that we are doing our best to regularly update with data as it becomes available. We also have a compatible user manual that is available upon request that will go along with all of these models. For more information, though, on the underlying technology, including fingerprints, Bayesian algorithms in general, the prediction scores, and visualization methods, I will direct you to these 3 papers. Thank you very much for your attention, and I'll flip it back over to Sean.
Thank you, Kim. We're already getting some questions, so keep them coming, and we'll definitely answer them at the end. I would just like to share a couple of additional slides that we have on the technology that I mentioned, as well as Assay Central. We've developed software tools for predicting the relative difficulty of synthesis for targeted analog molecules, as well as performing retrosynthetic analysis. And this first slide shows one of the tools that was developed by our team. And this example demonstrates some analogs for an antiviral drug called Tilarone that we've been working on, and as well as providing several synthetic pathways. Besides using standard bioisosteres, we have a large number of molecular transformations that are used, and these have been encoded so that we can identify modification sites on molecules and then automate the enumeration of potential analogs using our common medicinal chemistry approaches that are available to us. So in this case, I'm showing Tilarone as the first molecule on the bottom, and then one of the analogs that was proposed by the software. So this is a sort of an automated approach where we can put in a file of molecules, and then it will output chemically feasible molecules that could potentially be made, as well as providing some degree of scoring and availability. The next slide really speaks to the retrosynthetic analysis that we've been working on. And so far, we've used 3 primary methodologies to evaluate this synthetic feasibility. The first really involves fragmentation of known synthesized molecules that already exist and the relative presence of these potential fragments in the target molecule. And then the second method we've got really couples an automated retrosynthetic approach with this first method. And finally, we've got an automated retrosynthetic method that uses a rate of synthetic feasibility. And we basically have a separate application that we've created in addition to the previous slide. And we use a weighted mechanism that we've added to the algorithm as well to essentially penalize any molecular elements that are undesirable from a synthetic perspective. And we've leveraged quite a bit of experience from a medicinal chemistry realm in order to come up with these technologies. So you could imagine these being used to look at and come up with ideas of molecules that we could then feed into the Assay Central models as well. And we'd be happy to share more details on these technologies with you as well. Again, we've shown an example of Tillerone here being fed into the retrosynthetic analysis software, and then in comparison, the actual known synthetic pathway that's been recently published by an academic group. So there's quite a good degree of similarity. And we've undertaken this type of analysis with several molecules as well. So why choose our company? Why choose Collaborations Pharmaceuticals? I think, as you may have seen in the slides preceding this, that we're a pretty unique company. We do not have VC or angel funding as yet. We've been funded primarily through the NIH and DoD. To date, we've raised about $7.5 million in the last four and a half years. We also have a robust molecule and services pipeline, as I mentioned. So this gives us a little bit more flexibility to work on pretty much any project that we want to look at. And our pipeline pretty much represents that, in that we've been able to work on diseases like Ebola, Chagas disease, tuberculosis, HIV, as well as rare diseases like Batten disease, as well as several other lysosomal storage diseases that we're quite passionate about. So we have this flexibility to work on projects. And in addition, when we work on projects with collaborators, your IP remains your own. That's obviously important. And we just really focus on getting you the quality results that you need. So for example, our flexibility has allowed us to work on ADME and tox projects, most recently topics like drug-induced liver injury, which we've recently published on. And then most recently, as Kim said, we've been able to shift some of our resources to look at trying to address COVID-19 drug discovery. So I'd like to provide a few client use cases on this slide. We've obviously anonymized the companies because we're not allowed to mention them. And we've tried to keep this as broad as possible, but just to give you an idea of the type and diversity of the projects we've worked on. One of the projects we worked on with a company, a consumer product company, multi-billion-dollar company in the US, has been really on endocrine disruption. And we've worked on about 5 different projects with them over the last 3 years. We've been able to deliver suites of models around these targets. We've also curated data sets. I would say in aggregate, we've produced hundreds of models for them, looking at different ways to slice and dice the data that's available in the public domain. This has also led to at least 3 papers so far that we've put together. One has been published. It's also led to multiple talks at conferences over the last 2.5 years. A second example we have is another consumer product company in a slightly different segment. They were also interested in endocrine disruption, and we've been able to build models that were separate and unique for them as well, and provide those models to them. A third example is a rare disease company, again, $1 billion company based in the US. We've leveraged the data sets, relatively small data sets, for modeling ADME issues that they had, and as well prioritizing and designing molecules for them to synthesize. And we've fed data back through several cycles. This has led to a couple of projects with that company over the last year. And the final example, Company D, as we've called it here, is a small animal CRO company. They generate primarily in vivo data. We've been able to model animal phenotype data as well as target activity data. And we're currently on our 3rd unique project with them. So this gives you an idea of some of the types of companies at a very high level that we've been working with and the types of projects. What is a common thread between all of them really is the very close relationship we have with these collaborators, clients. And this obviously includes frequent interaction with them and sharing information and data and obviously models as we generate them. All 4 of these projects have utilized Assay Central as a core. And this is obviously something that we're happy to focus on. And if you have any projects that you think may be of interest, please get in touch. So just to give a brief summary of the types of projects that we can do, Kim has obviously mentioned what we can do on the machine learning side. Obviously, we have a heavy focus on data set curation. A lot of the time that Kim and her team spends is on this piece, putting together and cleaning up data sets. making sure that they're consistent, and then obviously validating the models that come out and trying to find test sets that we can use to do that. We have, besides Assay Central, which uses a Bayesian algorithm, we have multiple machine learning algorithms currently that are available to us. We have our own internal servers where we can do this. We have 10 GPU servers so that we can do deep learning models. We've done random forest, as well as about 6 other different algorithms. We've also published quite widely on algorithm comparisons. And we see in the future that we'll be integrating these algorithms more closely into the Assay Central toolkit. We can do projects where predictions may be needed for a particular target, whether that's an ADME or drug discovery target. And we can do some quite sophisticated cheminformatics analyses with different sizes of data sets. These models, as Kim mentioned, can obviously be used for both synthesis, prioritization, as well as testing. And we have the capability, as Kim had shown, to rank predictions as well. We can also look at chemical diversity of the molecules. Most recently, we've started to look at CNS permeability and see if we can come up with some new approaches that may add on to some of the algorithms that have previously been published. So this is an active area that we're currently working on. And we obviously think that there's a lot of need out there for these kinds of capabilities. And finally, as I mentioned, we've started to work and we have prototype software for calculation of synthetic feasibility and retrosynthetic analysis. So we're happy to welcome any new challenges that you may have, and we'd be keen to learn if there are any projects that you may be interested in working with us on. Obviously, the COVID situation has created a unique environment. We've been able to maintain pretty much full operation. We've basically half of our group has worked remotely throughout the pandemic. But obviously, we're working on computational technologies, and so we could pretty much do that anywhere. We would be happy to help you, and please pass on our information to any friends or colleagues that you have. I see that many of you that are joining us are actually well known to us and have been very good in sharing our information to other companies. So I'd recommend please do that. It helps us keep going. If you have any questions, as I mentioned, use the icon at the top of the screen, or if you want to put your questions on the attendee chat, that's also okay. We have a website that's got a lot more information, collaborationspharma.com, if you'd like more information on the types of projects, publications, or announcements. And both Kim and myself can be contacted directly by email as well if you want to do that privately. So that's pretty much it from myself and Kim. Thank you all for your attention, and we'll try to answer any questions. One other thing I would just like to say, this is the first webinar. We have several that we're planning to do, hopefully at monthly intervals, and we believe our next one will be focused entirely on our effort around COVID-19. So now let's answer your questions.